Hello, everyone. In this chapter, we are going to dive into sex linked inheritance. As we cover this chapter, we are going to try to understand the factors that contribute to whether you are a male or female, to distinguish the chromosomes X and Y. Now, this chromosome X and Y carry a number of genes. So we are going to address sex-linked genes. Also, we are going to look into sex-limited and sex-influenced traits. What does this mean? We are going to dive in that. We're going to look into X inactivation because, yes, the X chromosome is inactivated in some individual. We are going to address that. And finally, we're going to look at parent of origin effect. What I mean by parent of origin effect, it's whether you receive a feature from dad or from mom, the same feature is actually going to exhibit a different outcome, right? So now let's take a look at sex determination and sex chromosomes. In human, an individual who has the chromosomes X and X, these individuals are females. Homogametic sex means homo, same, gametic, gametes, right? Then individuals who are male are going to have an X chromosome and a Y chromosome. Now, the micrograph that you see here on your left-hand side actually is showing you the chromosome X here and the chromosome Y here. Now, look at these two chromosomes. These two chromosomes do not have the same size. X is much larger than the Y chromosome, which means that while in a female have homologous chromosomes, rigorously homologous chromosomes X and X. However, males do not have rigorous homologous chromosomes because they don't have the same shape. They don't contain the same genes. Yet, we say that they are homologous chromosomes. The only reason why we say that they are homologous chromosomes in males, it's because of the function, right? Because they are involved in sex attribution, but not because they are rigorously homologous chromosomes. I just want to throw it out there so you understand, why we call this sex chromosomes homologous chromosomes in spite of the fact that in males, obviously, they are not rigorously homologous. All right. With that being said, now, what determines sex development in human? Well, when we look at the development in a human, an early embryo has the potential to become male or female, complete potential, right? So the 5th week of prenatal development of an embryo shows an embryo that exhibit both males and female features, right? So when we look at this graphic representation of the level of development at embryonic level, what do we see? We see gonads, the future testes, right? And here we have the Mullerian duct, which is the future tubes that are going to lead to the uterus, the future uterus, right? And then we have the Wolffian ducts that are going to be the future vas deferens, all right? But at this point, while there is no differentiation, if the individual is going to become male or a female, the signal they receive later is going to determine at that point if the individual becomes male or female. More specifically, if the gene SRY is expressed because it is present, if that gene is expressed, what happens is that the Mullerian duct is going to be resorbed. It's going to be destroyed, right? While the testes are going to develop clearly and stand out. Eventually, the Wolffian duct is going to become the vas deferens or ductus deferens, all right? And we're going to have male features that develop with clearly the testes, the penis, the prostate, and everything else that make up male reproductive organs. Then, the other possibility is that the gene SRY is not expressed. In absence of the gene SRY, what will happen is that the Mullerian duct is going to develop, as you can see, while this Wolffian ducts are going to degenerate and the gonads are going to turn into ovaries, so that you have, at the end of the day, ovaries that develop, the uterus that develops, a vagina, the fallopian tube or the uterine tube that also develops. So really, everything depends on the signal that is received at a certain age of development of the embryo. So now, there was a time when we considered, when I say we, I mean biologists, those who are studying anatomy and physiology in human, we consider that, well, femaleness is actually the default sex. That was the overall knowledge, and this is also what is still being taught most of the time. Nevertheless, today it is now known that there is a particular gene, WNT4. The WNT4 gene actually is now clearly shown to be responsible for female organ development. Now, you may have absence of SRY gene, but if absence of SRY gene goes alongside with a mutation of WNT4, well, guess what? The embryo will not develop into a female. That's basically what we can see now. All right. So, sex chromosomes determine the gender. We have two types of sex chromosomes. We have the X chromosome and the Y chromosome. These two chromosomes are very different. How so? The X chromosome contains over 1,500 genes, while the Y chromosome contains only 231 genes. That's a huge difference. And this shows why, when we looked at the picture earlier, right, the micrograph, we saw the X chromosome being much longer than the Y chromosome. Makes sense. So the X chromosome is much larger than the Y chromosome. The X acts as an homologue to the Y male only because of the function, not because of the structure, but because of the function. So the Y chromosome does not cross over along all its length with the X chromosome, right? Many DNA segments that are repeated, these DNA segments that are repeated are called palindromes. They are called palindromes because they can be read from a particular from a particular area. It can be read in both directions, the same, right? That's what we call a palindrome. For instance, say I have a sequence that reads A, C, T, G, A. That's one part, right? The next part is going to read, let me see, T, C, A, G, T. Now, if we write the complementary strand, we are going to have T, G, A, C, T, A, G, T, C, A. Now, notice that this sequence here is the exact same sequence here. So this is a palindrome altogether. This is a palindrome, right? So many DNA segments are palindromes, and in addition, it may be destabilized. It may destabilize DNA replication. So now, let's take a look at the anatomy of the Y chromosome. The Y chromosome actually has a number of genes, but the most relevant genes I want to show you here is the SRY, okay, the PAR1, the bone development genes, the sperm development gene, AZF gene. So those genes that are shown here are all genes that are responsible for maleness in individuals, right? When these genes are expressed, this is when we see male features developing. Now, the SRY gene encodes a transcription factor. That transcription factor actually affects the expression of many other genes, including SOX9. It stimulates male development organ, sends signal for gonads to differentiate into male features, but also for the destruction of female features by programmed cell death. apoptosis, right? And it's going to stimulate the development of all organ structures, including the penis, the testes, the vas deferens, and so on and so forth. Testosterone and dihydrotestosterone, these two hormones, are secreted to stimulate the development of reproductive male organs. Now, there are some disorders associated with sex development. We have, for instance, hermaphroditism. Hermaphroditism, it's actually an old terminology, right, that is kind of still in use, but not as much as before, right? And this is to talk about individuals without a real differentiation, right, if they are males or female. So they are going to have male features and female features, but we cannot say if they are male or female. That's why some parents at some point had to choose which organ to keep and which organ to get rid of, which actually caused in the life of many people some trauma, because the decision was made that the individual is going to be male or a female in a very early age, while as the individual develops, we see that while the individual was made a female, some male features are appearing, and basically the individual not feeling a male. And the same thing is true for females, right? When an individual was hermaphrodite and a particular sex was assigned at birth, surgically, I mean. Then we have the intersex. The intersex, and it's when the genitalia are very much ambiguous. Ambiguous in a sense that, well, clearly there is no penis per se, there is no vagina per se. It's just there in limbo, right? And then we have pseudo-hermaphroditism. So in the case of pseudo-hermaphroditism, we have the presence of both female and male structure, but at a different stage of life. So in pseudo-hermaphroditism, we are going to have many cases, including androgen insensitivity syndrome. In androgen insensitivity syndrome, here we have X chromosome that blocks the formation of androgen receptors. And androgen receptors are receptors that can be that can receive a signaling molecule that is a hormone, right? That hormone can be testosterone, it can be estrogen. Now, testosterone is more abundant in male, estrogen is more abundant in female. So basically, any androgen chemical is a chemical that is associated with sex attribute, right? And receptors are going to be either testosterone receptors or estrogen receptors, just to mention these two. Then we have the 5-alpha reductase deficiency. In the 5-alpha reductase deficiency, this is when individuals are unable to convert testosterone into dihydrotestosterone. Okay? And the child has inside male anatomy, but looks like a girl in the outside. There are people today, they very much some movie stars, some influencers that appear on social media that actually are androgen, because the one I know of and the one I've seen, actually they look very much like ladies, but they are very much male in terms of the reproductive organs. So they have a penis and testes and everything. They don't have breasts that are developed, but their face is crazily like that of a lady, right? And the hair grows just that like that of a lady, right? That's a case of 5 alpha reductase deficiency. And then we have the congenital adrenal hyperplasia. So here, an enzyme is inactive. An enzyme is inactive, and because of that, androgens accumulate in the body. This causes actually precocious puberty in male or in female. Precocious puberty that can happen at a very early age, as early as 7 years old. So now here we are going to look into SRY gene. So when the SRY gene is present and active, because the gene can be present and not be active, right? So SRY gene is present and active, it turns on, it's going to cause the formation of testes, right? So with the formation of testes, the female structures are going to degenerate, right? Cholesterol, which is the initial substrate, right, is going to be converted into testosterone. Okay? It's going to be converted into testosterone, right? And that testosterone is going to attach to testosterone receptors. Okay? So we have some very specific receptors here. And by attaching to these receptors, we are going to have a differentiation, right? That differentiation is going to lead to the formation of epididymis, right? Duct deferens, the seminal vesicles, and the ejaculatory duct. Now, the other part of testosterone is going to transform into dihydrotestosterone, DHT. That dihydrotestosterone is going to have its own receptors, right? These receptors are going to cause the development of the urethra, the prostate, the penis, and the scrotum. All right? So you see that there are many features here that are involved in the development of a sex. So it's not just one single thing, it's many things, right? And if we have a deficiency in some of the features here, for instance, the 5 alpha reductase, well, we are going to have some aspect of maleness that are not going to develop, right? All right. So here we have some component of sexual identity. At a chromosomal level, we have XY that cause male to develop, X and X that cause female to develop, and this happens during fertilization, right? The gonadal sex are going to be indifferentiated until some point in the development of the embryo. And then beyond a certain point, so after 6 weeks, we are going to see some differentiation developing. Then we're going to have phenotypic sex that are going to develop, where we see external features that are noticeable, right? External features and internal reproductive organs develop into male or female depending on, which chromosomes the embryo received and the hormones that are going to develop, right? So this is we're going to start seeing some features clearly apart between male and female 8 weeks and after, right? And at puberty, we start seeing some features showing up. For instance, for female, breasts that are going to start developing. For male, hairy face, hair is going to start developing in the face. Okay, gender identity, it's basically the stronger feelings that are going to develop in male and female, right? And this is going to start in childhood and going to keep developing. And then we have the sexual orientation, which is the attraction to same sex or opposite sex that actually is going to start as early as from childhood. But really, this is not written in stone, right? This is not written in stone. All right. The sex ratio in a population, we are going to notice that there is a proportion of male to female in the human population. And that proportion of male to female in a human population is determined as follows. So we're going to consider the number of males divided by the number of female multiplied by 1,000. Okay, so now we have different levels of sex ratios. We have the primary sex ratio. The primary sex ratio, it's basically at conception. So we are going to ask the following. What are the individual at conception that are X and XY? Okay? So that would be the simple question. And then the next type of ratio is going to be the secondary sex ratio. At birth, what do we see at birth? Do we see a child with testes and penis? Do we see a child with vagina? Right? So we are going to be able to tell them apart there, right? But sometimes we are not going to be able to tell them apart, right? And then we have the tertiary sex ratio, which is at maturity. What do we see, right? So the sex ratio can change with age. It can reflect medical condition, and it can also reflect environmental factors, believe it or not. Now, let's take a look at the Y-linked traits. So these are genes that are associated with the Y chromosomes, right? The Y-linked traits are very rare, as the Y chromosome is very much devoted to maleness, right? To sex development. It is transmitted from male to male, okay? Because for an individual to be male, you have to have X and a Y. When you have an X and another X, there is no way you can be a male. There is absolutely no way that individual can be a male. Okay? And also, there are some Y-linked traits that can be involved in infertility, but are not transmitted, right? Now, the Y-linked traits have a certain pattern of expression. Patterns of expression on the X chromosome are going to differ from male and female. Now, remember that female are XX, males are XY. So female are going to give an X, and based on what the female is carrying on the X is going to determine what the male is going to express or not express, right? So it is going to be a cloudy, well, not a cloudy situation, but it's going to be a situation for males where, based on the X they receive from mom, where they're going to be called what we call hemizygous. Hemizygous because X does not match Y. So X is going to have some features that Y doesn't have, right? Female individuals are going to be perfect homologous. They're going to have perfect homologous chromosomes, so that actually the X-linked trait in female are just like autosomal traits, in a sense that the two chromosomes are so homologous that, they are going to behave in female as if they were autosomal traits, as if, right? Okay? While in males, the males are going to have a single copy from the X chromosome and no equivalent in the Y chromosome. Okay? And again, this is what we call hemizygous. Now, for a female, XX, for a male, XY. So basically, what is the chance that females are going to be developed from this gamete meeting this gamete, from this gamete from mom meeting this gamete from dad, where we have XX, that's female, and from this gamete from dad meeting this gamete from mom, we have XX, that's female, right? While for dad, this gamete Y, if it fertilizes the X from mom, we have a male, and the Y it fertilizes all the X from mom, we have a female. In other words, whenever dad gives the Y, the child is going to be a male. Whenever mom gives the X, whenever dad gives the X, the child is going to be a female. In other words, really, dad is the one determining the sex. Okay, whether dad is giving an X or a Y, it's what determines the sex. So, there are some traits that are carried by X chromosomes that are recessive, others are dominants. Now, whether the trait is recessive or dominant is going to tremendously affect individuals based on the sex, right? So now, let's take a look at how males and females are affected with X-linked recessive and dominant traits. Now, for an X-linked recessive trait, these traits are always expressed in males. Why? Because the X comes from mom that contains a recessive allele. Dad has a Y, giving a Y has no equivalent in X. Therefore, that single copy of the recessive trait is going to be expressed. Okay? Now, for the X-linked dominant trait, it's going to be expressed in female in as much as there is one copy. It's going to be expressed in female, but it's also going to be expressed in males. However, what is worth mentioning here is that a dominant trait that is carried by X is going to be more severely expressed in males than they are in females. Let me repeat that. The X-linked dominant trait are going to be more severely expressed in males than they will in females, right? Now, the X-linked recessive traits are going to be expressed in females that are homozygous, right? Homozygous recessive, okay? Very rarely in female that are heterozygous, okay? Because you have a dominant allele that is going to be expressed there. Now, the affected males are going to inherit traits from heterozygous or homozygous mother. Okay, think about it. Heterozygous or homozygous mother, because a mother is going to be like this: X lowercase a, X lowercase a, or X capital A, X lowercase a. So giving this one, mom is going to cause the child to be affected, right? Giving this one, mom is going to get the child to be affected, but giving this one, mom is going to get the child to not be affected, right? So this is heterozygous and this is homozygous, right? Affected female are going to inherit the trait from, I'm talking about the recessive trait, right? They are going to inherit the trait from affected father and affected mother, right? Whether they are heterozygous, right, or not, okay, or not in the sense that they are homozygous recessive, right? So for the dominant, in addition to what we already mentioned, we have that we're going to have a very high rate of miscarriage due to early lethality in males. Remember that the dominant allele that is carried by X is more severely has more severe impact in males. Therefore, the level of lethality is going to be higher in male. Now, the dominant allele is going to be passed from male to all daughter regardless, but never to a son, because a father has an X and a Y. The Y doesn't have any dominant feature. So therefore, Males can never receive that trait. All right. So the X-linked recessive traits, these are recessive traits that are expressed in female only if two copies are present. Okay? Only if two copies are present. Now, if the condition does not cause the death of the developing embryo, right, a male may be healthy enough to transmit it to the offsprings. Okay? Let me repeat that. If the condition is not lethal, then the chance that dad is going to transmit it to its offspring is high. Okay? So basically, the condition has to not be lethal for that to pass it to the offspring. Okay? For some examples are going to be ichthyosis, which is a case of dryness of the skin, right? The skins are kind of scaly, right? Here we have an enzyme that is missing. That enzyme that is missing is unable to process cholesterol from the skin. As a consequence of that, the skin is very dry. Another example is color blindness. About 8% of male of European ancestry have the condition of color blindness. All right? And this is caused by a recessive gene that is on the X chromosome. And then we have hemophilia. Hemophilia, it's a blood disorder by which the individual does not know how to blood clot, right? And so here we have the example of ichthyosis. And that ichthyosis, you can see that mom is unaffected, dad is affected, right? Now, the daughter here is a carrier, but not affected, right? But take a look at this. The son here is affected, okay? So only males are completely affected while females are not affected. The most that can happen is that they can be carriers. All right, so now in terms of hemophilia, here, when we look at the case of hemophilia, in this particular pedigree here, we're going to start with Queen Victoria of England. So Queen Victoria of England was suspected to be a carrier. She didn't have hemophilia, but she's suspected to be a carrier. So Queen Victoria had a number of children, including Leopold, Duke of Albany, Alice, and many other kids here. But I'm not going to go into those. The one I'm going to address here is Alice. Alice, the daughter of Queen Victoria, gave birth to a number of children, including Alexandra here. Okay? Alexandra is a carrier. Her mom was a carrier, right? But Alexandra, after marrying the Tsar, Nicola of Russia, they had a boy called Alexei. Alexei actually developed hemophilia. And Alexei was the only son of the Tsar. And what happens is that because he was the only son, it was very important for the Tsar actually to make sure that his son was healthy. So he developed some relationship with a guy called Rasputin. Maybe you've heard of him. Rasputin. And Rasputin claimed that he could cure Alexei with hemophilia. So the Rasputin actually became very influential and caused the Tsar to make bunch of political mistakes. Political mistakes that actually cost him the throne. And this is how the reign of the Tsar Nicola actually ended, because of the bad advice he received from Rasputin, and he had a grasp on Nicholas because of what he claimed that he could do for his son. And at the time, it was very important to have a son who would inherit the throne after dad. Anyway, I wanted to kind of tell a little bit that story. All right. So the X-linked dominant traits are going to be relatively rare because, remember, for male, these traits are going to be very severe and most of the time are going to cause lethality. Okay? So now, something that we need to mention here is that gene expression actually differs between sex, whether you are a male or a female. A female who inherit a dominant X-linked allele, or in whom the mutation originate has the associate trait, right? But the male who inherit the allele is going to be usually more severely affected. And the reason why they are very severely affected is that there is no other allele on the Y chromosome to balance anything out, right? Which is a major issue for the dominant allele. So most cases of the dominant allele are going to be the result of de novo mutations. These are new mutations that occur that were not there before, right? Now, some examples of these cases are the Rett syndrome. The Rett syndrome, it's actually a condition in which a baby is born normal, but then start developing some defects such as difficulty swallowing, difficulty having coordinated movement. The individual grows and become completely unable to live an independent life. The disease is a neurodegenerative disease. It's a muscle disease. It's many different systems are affected in these individuals, right? And then another example is incontinentia pigmentis. This is pigmentis. This is when the skin color is not the same everywhere. It's not the same everywhere. You have some patches in the body that are going to develop some redness, maybe loss of skin color, and so on. So this is basically what you can see here, right? Incontinentia pigmenti. Now, how can we actually show how this disease is inherited? Say you have a female here that is heterozygous, right? Capital R, lowercase r, carried by X. The oocyte are going to be capital R, lowercase r. If that lady is has a child with a man who is unaffected, so lowercase r, that unaffected male has the chance to have affected daughter here, unaffected daughter here. Then we have unaffected son here and an affected son here. So we see that we have 50% of daughters have chance to be affected, 50% of daughters chance to not be affected. For boys, 50% of boys have the chance to be affected, okay? And 50% of boys have the chance to not be affected, right? This is for mom being heterozygous, right? That we have these ratios. Now, when we want to solve problems with X-linked genes, how do we go about this? Well, the way we are going to go about this is to first of all look at inheritance pattern. Look at inheritance pattern, right? So basically we are going to take a look at the parents and offspring and try to connect individuals and see the particular feature we are tracking, just like the way we did with autosomal disorders, right? Whether they are dominant or recessive, right? So that's what we are going to do here. However, the only major difference here is that as we are looking at the pattern of inheritance, we have to pay close attention to the sex, right? What is the proportion of males and females that are affected, right? We are going to draw a pedigree. We're going to list genotypes and phenotypes, and also, if possible, their probabilities. Now, knowing the genotype, we can come up with the probabilities, right? So then after listing the genotypes and phenotypes and their probabilities, we will now assign genotypes and also phenotypes of the parents, right? Once we do that, we're going to determine how alleles are going to form, right? What are the different possibilities of alleles for the parents? And then we are going to unite this gametes, right? We're going to bring the sperm cell and the egg cell. We consider the alleles that the sperm cells and egg cells are carrying, right? We do that in a Punnett square, and then we are going to determine the phenotypes and the genotype ratios, right? And that for the F1 generation. And as we are done with that, then we are going to predict for the F2 generation and F3 generation and so on and so forth. But we have to follow the same steps as we go from one generation to another generation. More specifically, we have to assign genotypes and phenotypes for parents. 2, we have to determine how alleles are going to separate into gametes. And finally, we're going to not finally though. We're going to unite the gametes in a Punnett square to determine what is the ratio of genotype and the ratio of phenotypes. All right. So the traits that affect the structure or function that is going to be present in one single sex is going to be called sex-linked trait, right? Going to be called sex-linked trait. So these genes can be carried by the X-linked, okay? They can be carried by autosomes. No, let me step back here. Let me step back here and make sure that I'm not saying anything confusing. So the traits that affect a particular structure or a particular function, and that is present in only one sex, say a female, these are going to behave as if these traits were autosomal traits or X-linked. There we go. That's what I wanted to say. Okay? So, a good example of this, it's going to be the beard growth. Right? The beard growth, it's actually carried by sex chromosome. And in as much as that that feature occurs only in one sex, then it doesn't matter really that this I'm getting confused. Then it doesn't really matter that this gene is carried by an autosome or a X-linked gene, right? The milk production, it's very specific to female, okay? While the preeclampsia in pregnancy is also going to be very specific to female. So in that case of preeclampsia in pregnancy, this is a case of a gene from the father that may affect the placenta development, right? The placenta development and affects actually the blood pressure in a woman, okay? If something is not confusing here, please let me know. I mean, if something is confusing here, please let me know, okay? Because sometimes the way things come out, I may think it's clear, but maybe it's not clear to you. Please let me know. Now, sex-influenced traits. In sex-influenced traits, the genes may be autosomal or X-linked. But very importantly, the sex is going to influence how the trait is going to be expressed. Okay? Whether you are a male or a female is going to affect how the feature expresses. And this is caused by hormones that are different from one sex to another. Males have a bunch of testosterone, females have a bunch of estrogen, which obviously are different, right? So in human, for instance, patterns of baldness, right, is very much a problem that by the sex, right? A heterozygous male is going to be bald, but a heterozygous female will not be bald, right? The response to some treatment, okay, for instance, the glioblastoma, all right, female patients are going to live longer than male patients after treatment, okay? And that because of just simply the sex, okay? Now, let's talk about X inactivation. In X inactivation, females, we know they are XX, right? And we do know that in the 2X, one is going to be inactivated. And why is one of the X in female going to be inactivated? It's simply because there is a matter of dosage of expression, right? There is a matter of dosage of expression. A male has only one X chromosome. A female has two. For the female to not have a too high of an expression of a particular trait, one of the X's is inactivated. Okay? We are going to address this into more details. Now, that's one. The second thing, it's that there is in a particular, in a female that is heterozygous, for instance, we are going to have a different expression of alleles based on which X is inactivated and which X is activated, right? And because of that, heterozygous female are going to have what we call mosaic expression. Okay? A female who is heterozygous is going to have a mosaic expression. In other words, we are going to see two phenotypes in the same individual, right? There is a particular gene that is called XIST. That XIST gene encodes an RNA. That RNA binds to an X chromosome and cause inactivation of that X chromosome. That is the reason why, in spite of the fact that a female is XX, one of the chromosome is going to shut off, while the other one is what the individual is going to rely on. Now, here is the thing. The one that shuts off in one cell is not the one that shuts off, the same that shuts off in another cell. Okay, let me repeat that. The one X chromosome that shuts off in one cell is not the same chromosome that shuts off in another cell in a female. Okay, that is very important to remember. So that inactivation occurs in early prenatal development of a female. The adult female is actually going to have patches on their body, right, that differ basically based on which X is being expressed and which X is not being expressed. At the end of the day, even though the genotype is the same in an individual, the phenotype is going to be different. Let me repeat that. At the end of the day, even though the genotype is the same in an individual, the phenotype is going to vary. Okay? So now, because the genotype essentially is the same in the whole individual, and that the phenotype is not the same, what happens is that this is going to be an example of epigenetic change. That epigenetic change can take different forms, including the addition of what we call a methyl group, which is a functional group. That methyl group is going to change the pattern of gene expression. In other words, which gene is being expressed where. And that inactivation, that methylation, is going to affect not only the X chromosome, but also it is going to affect a number of other genes that are not necessarily attached on the X chromosome. Okay? But an epigenetic change, it's a change on the genetic information that is not associated with the change in the sequence of DNA. So the sequence of DNA, it's exactly the same in all cells. However, the pattern of gene expression in those cells is altered because of the addition of a methyl group or the addition of another functional group that otherwise affect the way the genes are going to be expressed. So here we have the case of a mosaic in a female. So we have here a maternal X and a paternal X, right? So we have a fertilization of the egg. Actually, we have the fertilization of the egg. So now we have this zygote, okay, that develops cells. And now these cells are going to undergo cell division, okay? So now, as the cells undergo cell division, now we have in this individual here, one of the X that is going to be inactivated. Okay? One of the X is inactivated here. One of the X is inactivated here. Now, which X gets inactivated, it's not predictable. So we don't know if the X you receive from dad is the one that is going to be inactivated, or if the X you receive from mom is the one that is going to be inactivated. It is completely random. Because it is completely random, and because different cells are going to inactivate different X, we have actually a mosaic of cells here that are all different. Well, all different. Not completely all different, but like this group is different from this group, right? So that when we look at the tissue, we see actually some cells that have a certain color, some cells have a different color, okay? So here we have the skin without the normal sweat gland, in this case, and the skin with a normal sweat gland. So that in this individual, the sweat glands are not equally distributed throughout the body and throughout the skin. The sweat glands are not equally distributed throughout the body and throughout the skin, just because of that mosaic, right? So some parts of the body are going to sweat more than other parts of the body. That's the bottom line, right? All right. So X inactivation can actually be used to check the sex of an individual. The nucleus of a cell of a female is going to have a bar body. The bar body, it's actually a X inactivation. So here, this is a bar body, this is a bar body, and this is a bar body. So all the sex that are inactivated are bar bodies. Okay? The consequence of X inactivation on the phenotype can be very patchy, in the sense that homozygous X-linked genotype, homozygous X-linked genotype is not going to have an effect So you are not going to see a mosaic, if you will, but the heterozygous is going to give you a mosaic, okay? Because, well, you have two different alleles, and because you have two different alleles, the phenotype at the end of the day is not going to be the same because everything depends on which allele is being expressed and which allele is inactivated. A good example of X inactivated inactivation phenotype is going to be in the calico cat. In the calico cat, when you look at the fur, we have some areas that are orange, some areas that are black, okay, are actually some good example of what is being expressed where and what is not being expressed, right? So a female is going to express this orange fur when the pigment orange is the one that is active, while the black fur is going to express when the allele that expresses melanin is the one that is expressed. So basically, here the gene involved if in melanin production are not expressed here, so that the phenotype orange is the one that gets expressed while it is expressed in this area. Okay, so you have a mosaic basically on this calico cat. So that's in heterozygous. All right, so in human, X inactivation can be used to identify carriers, okay, for some X-linked disorders. A good example is the Huntington syndrome, right? Also called the mucopolysaccharidosis. Mucus, right? Polysaccharide. So basically here you have a lot of sugars, complex sugars, in the mucus of people with Hunter syndrome. Then you have the Lesch-Nyhan syndrome. Man, I'm drawing a blank. I don't remember what that is. I just read about it, but I'm just drawing a blank. Then we have the Rett syndrome, which severity is going to vary from one individual to another based on what X is being actually what X is being inactivated. So unequal X activation pattern can occur if the two chromosomes have different alleles. We saw that, right? For a gene that controls cell division, that's a major issue, right? That's a major issue because now we are going to see a difference in survival based on basically which X is activated and which X is inactivated, right? So some cells are going to have a survival advantage while others are going to have clearly a disadvantage. Now, how about the parent of origin effect? Parent of origin, father or mother. So in Mendel experiment, it didn't really matter if someone was a father or someone was a mother. Now, in the case of Mendel, we worked on plants, right? It didn't matter if a plant was female or if a plant was male. We did really focus exclusively on the threat. Is it there, yes or no? Or do we have some, you know, is the threat there, yes or no? So for some genes, the parent of origin, whether they are dad or mom, is going to influence the threat. For instance, for males, when you reach a certain age, there are some issues that are likely to happen in the offspring than they would in young folks, right? We have an age onset, right? And symptom severity is also going to be influenced by, did you receive the feature from mom or from dad? Now, there is a mechanism of parent of origin effect. That mechanism is called genomic imprinting. Genomic imprinting, it's basically when some parts of the genome are not expressed, not because there is a defect in the genetic sequence, but because there is a slight modification on the genetic molecule, right? We talked about epigenetics, right? We mentioned that earlier. That epigenetics, actually, it's, for instance, the addition of a methyl group. That methyl group is going to affect the way genes are expressed. Now, from one generation to another, we may see a difference in gene expression. A gene that is imprinted in one particular generation may not be imprinted in the next generation, right? It can vary. And the reason why it varies, it's because there is a change in the pattern of imprinting during the process of meiosis. Okay? Specifically during interphase, you have to have the genetic information to be copied. And then that genetic information has to be distributed, right? There are some conditions that are incompatible with some events of cell division. For instance, replication, right? Methylation is not compatible with replication, so these methyls have to go away. Okay? That is the reason why during meiosis, well, you have removal of methyl groups, so that where are these methyl groups going to come and bind and attach again, there is no prediction whatsoever as to which area is going to be imprinted in the next generation. So also something that is very worth mentioning, it's that the developmental timetable is not the same for sperm cells as it is for egg cells, right? So here we have genetic imprinting with addition of a methyl group here. The addition of a methyl group here is going to interfere with gene expression, okay? So genomic imprinting can be caused by methyl group addition, right, on a gene, which is going to cause a gene to not be expressed. Now, for an imprinted gene, the copy inherited from either the father or the mother is always covered with methyls, even in different individuals. Let me repeat that. For an imprinted gene, the copy inherited from either the father or the mother is always covered with methyl, even in different individuals. So this means that there is a track record at a cellular level as to which gene is being methylated and which gene is not being methylated. Okay, let me repeat that. There is a track record of which gene is being methylated and which gene is not methylated. Okay, so some examples of genomic imprinting are the central precocious puberty. This central precocious puberty is always inherited from the father. And actually, this is caused by a gene called the MKRN3 gene that is again inherited from the father, that when covered with methyl is going to be imprinted, and as consequence of that, the girl is going to reach puberty at a very early age, as early as before 8 years old. Okay? So remember epigenetics, right? Epigenetic alteration, it's that alteration of the genetic information that does not affect the sequence of DNA. Does not affect the sequence of DNA, but it does affect gene expression because of some residues such as methyl groups that are attached to genes. Okay, so imprints are erased during meiosis. I already mentioned that. Imprints are erased during meiosis. So let's take a look at our cells here. So here we have a gamete, an egg cell, right? And a sperm cell here. So these two cells are brought together through a process of fertilization. So now we are going to have now a zygote that develops, right? And that zygote that develops is going to have here female, okay? Here a male. So now in somatic cells, this is what we are going to have. In sperm cells, in reproductive cells, future sperm cells and future egg cells, so we are going to have the same thing here. However, during meiosis, notice that what was imprinted here and here is not imprinted in here anymore or imprinted in here. So the old imprints are erased, right? That old imprints that is erased, right, is going to be restored. Well, restored. Let me cancel that word restored because it's not really a restoration, right? So imprint is removed, and then we have some new imprints that develop. Now, the pattern of imprintings that develop is not predictable in a sense that see here where the imprint is happening here and where the imprint is happening here, so that the oocytes that are imprinted and the sperm cell that are imprinted don't have the imprints at the same location. They don't, right? And this is made possible because during meiosis, we have a removal of the imprints. Okay? So, what is the importance of genomic imprinting? Genomic imprinting is going to become particularly important during the development of an embryo. Okay? So, during the development of an embryo, some parts are going to start differentiating to develop some organ systems, while other parts will differentiate into some different organ systems. In early stage of development, there are some genes that are turned off. They are turned off because of imprinting, and they don't get turned on until a late stage in embryonic development, right? But those genes that are going to be turned off are going to be turned off because of imprinting. Now, interestingly though, we notice that the imprint is going to occur in genes that are organized in clusters. So we're going to have clusters of genes that are imprinted. But what I need also to mention is that some genes are not going to be expressed not because they are imprinted, but by virtue of where they are located. In other words, when they are located close to areas that are imprinted, these genes tend to not be expressed. Okay, which actually adds another layer of complexity in gene expression. So, one gene in a cluster could be essential for early development, other become imprinted due to bystanders. So, basically, bystander effect. So, basically, well, these genes technically are not imprinted, but because they are in a cluster, they cannot be expressed because of that cluster of imprinting, right? Maybe, for instance, in the middle of it, right? So most of the genes that are all around are not going to be expressed, but virtue of being in the middle of all these genes that are not going to be expressed, that gene also is not going to be expressed. All right. So it takes two opposite sex parents to produce a healthy embryo. It takes two opposite sex parents to produce a healthy embryo. So the male genome controls placenta development, while the female genome is going to control the embryo development. So you understand that if the male genome is not expressed, that is a problem, right? The placenta will not be produced, while if the female genome is not expressed, well, we're going to have a problem with the development of the embryo. Genomic imprinting may also explain the reason why we see incomplete penetrance. Okay, maybe a concern in assisted reproductive technology. Why? Because in assisted technology, actually, we don't see, you are isolating you are isolating cells and taking cells out of context. And these cells that are taken out of context are not influenced anymore by both environmental issues, environmental context, and the hormones, right? As consequence of that, well, all the imprintings that need to develop may not develop properly, which means that at the end of the day, the embryo actually can be affected as to how that embryo is going to develop. So, what are the relationships between imprinting and human diseases? So humans are going to have a number of genes that are imprinted, about 150 genes that are imprinted, if not more. Okay? And at least 60 of them affect the health if abnormally expressed in an embryo. There are two particular syndromes that we are going to mention here. Two particular syndromes that are caused by genes that are carried by chromosome 15. So there is a deletion in chromosome 15 that, based on who the child gets that deletion from, is going to develop one syndrome or another syndrome. Now, if a father is the one giving chromosome 15 with the deletions, this is going to lead to a Prader-Willi syndrome. That Prader-Willi syndrome, it's basically when a child is born and is unusually obese. There are many symptoms, obviously. One of the symptoms is obesity. That doesn't happen in all individuals. Small hands, that doesn't happen in all individuals. The distance between the eyes and many other features, phenotypical features that are typical to the Prader-Willi syndrome. Now, this is when the defect is received from the father. But if the same deletions on chromosome 15 are received from a mother, now the child is going to develop what we call the Engelmann syndrome. The Engelmann syndrome, it's basically a condition that affects the development of an individual. Okay, with, in terms of nervous system development, there is an issue. The coordination of movement, it's an issue. Learning, it's an issue in those individuals. And then the abnormal imprinting can be associated with a number of health conditions, including diabetes mellitus, autism, Alzheimer's disease, schizophrenia, and it was shown recently that there is a possibility that this can be associated with male homosexuality. So here are two children, one with the Prader-Willi syndrome, okay, who received the chromosome from dad, and the other one with Engelmann syndrome, who received the same chromosome with the same deletion, this time from mom, right? All right, so finally but not least, differences in development of sperms and eggs can explain some parents of origin effects. Like, for instance, the Huntington disease. We already talked about the Huntington disease, right? The Huntington disease is going to have an onset at younger age with fast and more severe symptoms if inherited by a father, while this is much slower, right, if inherited by the mother. The Noonan syndrome, it's going to be expressed in children uh, with a parent, with a dad who is actually old in age. Okay, so as the age risk, I mean, as the father grows old in age, like in the 70s, because at 70, a male can still have children, right? So being old in age for male is an increasing risk for having a child with Noonan syndrome, which is basically very, not very common syndrome, but a syndrome with developmental issues. So that's about what we have for this chapter. If something sound confusing, please let me know. Send me an email, and I'm going to clarify anything that you may struggle with. Thank you for your attention.